Without further ado, let's go to our session. Dr. Rafiq is a busy lady with several important affiliations. She is the program leader for CAR-T Basic and Translational Research at Winship Center for Cancer Immunology. She is also assistant professor of hematology and medical oncology at Emory University School of Medicine. I hear she also keeps her brilliant graduate students very busy with exciting projects. Today, we will focus on one of the high-impact projects from the lab, research done on CAR T-cell manufacturing with a multi-cytokine backpack platform and the insights into CAR T-cell metabolism they were able to gain in this work. Before we get into her presentation, I would like to introduce very briefly the 908 Devices Rebel device. The device was used along many other analytical assays and methods to gain some of the insights in this project. From spent media analysis side, the work to gain insights into T-cell metabolism was done with the Rebel. Now, this was only possible as the Rebel requires such a small amount of sample. And with this analysis, each sample run, we get the concentrations of full list of analytes in the panel, including all amino acids. The results are done in about 10 minutes per sample, so as here, we used 96 well plate format for the samples. The sample preparation as well as the device operation is easy, which means that we have a feasible way to gain these insights and to give further direction to further developments. Now, highlighted here are just some key findings from previous literature on why amino acids matter and why including spent media analysis might provide you with more insights into your process. Choose your favorite amino acids. Which one matters to you the most? These were just the highlights. With the Rebel, we can do sample spent media analyte level comparisons in freshened spent media, as shown here, or, of course, look at analyte levels in a time course, following along the consumption, as shown here, or accumulation of key nutrients such as amino acids. And with that, I would like to get us to our session. Dr. Rafiq, please take over and let's hear some great insights into your work.
Thank you for that introduction, Mila. I'm excited to be here today to discuss our project on engineering functionally improved CAR T cells with the multi-cytokine backpack platform and provide some insights into metabolism. As Mila mentioned, my name is Sarwish Rafiq. I'm an assistant professor at Emory University School of Medicine, and I'd like to thank 908 Devices for giving me the opportunity to present some of our work today. So I'm going to first give a very short, brief overview of chimeric antigen receptor T cells, or CAR T cells. So CAR T cells are T cells that are gene modified to express synthetic receptors, and these receptors are designed to mimic a T cell receptor in which they have a binding domain, generally derived from an antibody as a single chain variable fragment, a hinge, a transmembrane domain that anchors it in the membrane, and then intercellular signaling domains that activate and provide co-stimulation to the T cell. This is a way in which we can redirect T cells to recognize and eliminate cancer cells. So there's been a lot of rapid clinical progress in the field of CAR T cell therapy. So some of the first patients were treated first in 2010, if you look at this timeline. And, from 2010 to last year or, this year, we now have 6 total FDA-approved CAR T cell products. And the majority of these are in hematological malignancies and where they do fairly well, but there's still a lot of biological and manufacturing challenges that remain that we need to address in order to expand upon the utility and the usability of these therapies for more patients. These are the 6 currently FDA-approved CAR T cell products, and you can see they come from a variety of different companies, and they have pediatric and adult indications, but mostly for leukemia, lymphomas, and multiple myeloma. They are manufactured generally using anti-CD3 beads or anti-CD3 antibodies, along with IL-2. And they have a wide range of response rates, some of them having very high complete response rates across different malignancies. However, most patients that receive a CAR T-cell product don't achieve a sustained complete response. And so if we look at this figure here, we can see the different types of leukemias and B-cell leukemias and lymphomas that CAR T-cells are currently approved for, and you can see each one of these figures represents 10% of patients. So about 10% or less than 10% of patients will have a manufacturing failure where they are not able to produce a CAR T-cell product. After infusion, depending on the malignancy, a subset of patients will have primary resistance to CAR T-cell therapy. Patients that do achieve a complete response, many of them will relapse either with antigen positive or antigen negative tumors. And so the number of patients that actually have sustained complete responses or durable sustained complete responses is low. One of the insights that's currently being explored from correlative studies is that the initial CAR T-cell product phenotype can greatly impact therapeutic outcomes in patients. And so this figure is from the ZUMA-1 study, in which they saw that durable responses were associated with less differentiated CAR T-cells in the product that patients received. And so this and other studies have indicated that limited stem cell memory cells, or CD8 cells, in proportion to tumor burden are associated with failure. Patients who achieve CR have higher frequencies of CD8 T cells expressing memory signatures in their CAR T cell product. CD8 T cell exhaustion signatures were associated with failures to achieve responses, and then products containing large percentages of central memory or memory stem cells have superior expansion, anti-tumor activity, and persistence in patients. Furthermore, recent studies have indicated that these naive or memory CAR T cells may also have decreased toxicity in patients. And so we know that there are significant toxicities such as cytokine release syndrome and neurotoxicities known as ICANS that are associated with CAR T cells. And both preclinical and clinical studies using either sorted or enriched products for naive or memory CAR T cells have indicated that these products may result in less severe toxicities being experienced by patients. So with that, let's talk a little bit about how CAR T cells or autologous CAR T cells are currently manufactured. The majority of these products, a patient comes into a clinic and gets their blood drawn. These cells are then sent to a manufacturing facility where the T cells are isolated and activated either with CD328 beads or CD3 antibody. IL-2 is added to activate and expand the cells, after which they are genetically modified using retroviruses or lentiviruses, which encode for the chimeric antigen receptor. Once the T cells are expressing the CARs, they're expanded and then packaged and delivered back to the patient as a form of highly personalized medicine. And once infused back into the patient, they are a living drug in which they recognize the tumor cells, can secrete factors to kill the tumor cells, produce an inflammatory response, and expand. We also know that this manufacturing process can impact the product phenotype. And so many different steps along this way can cause differentiation and exhaustion in the T cells. And this includes the activation step, in which we're using strong activators of the T cell, and also the gene modification step, and finally the expansion step, where we're using gamma chain antibodies to expand these T cells, which are known to cause more differentiation and exhaustion. Knowing this, we worked with a collaborator, Dr. Eric Dreden at Georgia Tech, and he had a technology in which he could rapidly assemble and screen multivalent cell-directing therapies for leukemia. So his lab was exploring ways in which antibody-coated particles could bridge T cells and tumor cells and also deliver cytokines to the T cells to improve their cytotoxic potential. And their lab referred to these as cytokines. And so this was an interesting way to deliver cytokines and bring T cells and cancer cells together. And as you can see from their publication here, the cytokines are labeled in orange, and they're found where the T cells and the cancer cells are meeting, demonstrating that they're actually bringing these two together and providing additional support with the cytokine. We postulated we could possibly use this technology but pivot it for CAR T cell manufacturing, in which we could provide activation and co-stimulation similar to current technologies, but also provide the cytokine support that is needed for CAR T cell manufacturing. So as a proof of principle, we first assembled particles that looked similar to a Dynabead. So they had CD3, CD28 antibodies, but then had an IL-2 antibody in which we loaded IL-2 onto these particles. And so here we can see that this particle can provide activation, co-stimulation, and IL-2 support to the T cells. The particles could be manufactured in a consistent way in which we saw similar sizes of particles and protein coding from batch to batch. Furthermore, once we used these particles to manufacture CAR T cells, we saw that we could circumvent the need for using exogenous IL-2. And so if we use the traditional methods of Dynabeads plus IL-2, we get good CAR T cell CAR expression on T cells, but we could also get equivalent expression using this cytokine backpack approach. And furthermore, we got similar transduction, and if we added more IL-2 to these cultures, we could see that that didn't provide any further transduction efficiency, indicating that the IL-2 that the backpack, which we called multi-cytokine backpacks, was sufficient for the manufacturing process. We can see that this is also the same with free antibodies versus our multi-cytokine or MCB, where we got similar CAR transduction speeds between these two different processes. And then in terms of cell numbers, we also got equivalent cell numbers. So there's a non-significant difference between the Dynabead and our multi-cytokine backpacks. And if we add exogenous IL-2, there's a non-significant difference between the amount of T cells that we produced at the end of manufacturing. So having this platform was important, but we wanted to expand upon it and find an ideal manufacturing reagent that would produce CAR T cells with superior phenotypes. In order to do this, we generated a library of different multi-cytokine backpacks that provided different signals to T cells during CAR T cell manufacturing. And so the signals that we studied were anti-CD3, anti-CD28, IL-2, and IL-15, which has been demonstrated in the literature to produce less differentiated CAR T cell products. And so how we went about doing this is we had our iron oxide microparticles, which we could magnetically remove from culture after manufacturing. We would incubate these with antibody or antibody fragments, and then we would load these cytokines onto these antibodies to create our multi-cytokine backpacks. And so our workflow was such, after creating this library, we did a 96-well format manufacturing process in which we produced CAR T cells in a 96-well format and then studied them through high dimensional throughput flow cytometry and spent media analysis to identify multi-cytokine backpack formulations that would produce CAR T cells with superior phenotypes. And so first, we wanted to make sure that our library that we engineered was diverse. And we can see with this 3D-dimensional plot with IL-2, CD3, CD28, and IL-15, we get a wide array of expression of these four molecules on our library particles. So through our library screen, and we manufactured CAR T cells, we first looked at transduction efficiency and how much CAR these T cells were expressing. And so if, just to orient you, this is looking at percentage of CAR T cells. Our MCB formulations are across the bottom. So each of these numbers represents a particular formulation of our MCB. And it's organized from lowest expression to highest expression. The dotted line here is our CAR T cell transduction that we achieved with the control of Dynabeads plus IL-2. And so you can see this is a very shortened axis. So we were able to essentially achieve similar transduction efficiencies as Dynabead plus IL-2 with our MCBs. It's important to note in this that during this entire process, we are not adding exogenous cytokines of any sort. And so all of the cytokine support that these cells are receiving are from the MCBs directly. So next, if we look at less differentiated phenotypes, we can see that there's certain formulations that have improved or have larger percentages of less differentiated T cells in their final product in both CD8 and CD4 CAR T cells. To further dive into which particles would be the best, we looked at the relationship between which signals were associated with less differentiated CAR T cell phenotypes. And similar to what we expected based on our literature search, we found that beads with higher levels of CD3 and IL-2 were negligibly correlated with less differentiated phenotypes, and beads with greater percentages of CD28 and IL-15 were positively associated with these less differentiated phenotypes. Next, as I mentioned, within our library screen, we used multidimensional flow cytometry in our 96-well format, but given the format of how we manufactured these, it lent itself very well to doing spent media analysis with the Rebel machine. And so as a very small amount of supernatant was needed for these analyses, we were able to harvest supernatant from our screen and send it through the Rebel for spent media analysis. So first, if we cluster the results from our flow cytometry analysis, we can see that if we have our CD4 and CD8 less differentiated CAR T cell here, and we look at clusterings of low expression versus high expression, we actually do get 3 distinct culture clusters of particles that produce more differentiated, less differentiated CAR T cells, and less differentiated CAR T cells. And it's important to point out here that actually the Dynabeads plus IL-2 is within this poor producer cluster. So using these 2 clusters, we plotted our spent media analysis, and we can see that they actually cluster significantly differently in the nutrients that they're using within the media during manufacturing. And so we could see that the particles that cluster in the superior CAR T cell products have more of certain types of amino acids left and have used different amino acids than the poor performing MCBs. Using this data plus additional clustering analysis in which we added in the ratio of CD8 to CD4s, we were then able to identify MCB particles in which were consistently better in all of these analyses. And so we're looking at 27, 43, 9, and 26, and these were the ones that were consistently performing better within all these analyses. Therefore, we decided to move on to further functional assays using these 4 different MCB particle formulations. As a reminder, the multi-cytokine backpack is providing the anti-CD3, the anti-CD28, IL-2, and IL-15 to the T cells in a all-in-one reagent. And so interestingly, if you look at the formulations of the best performing MCBs, we had formulations that had CD3 and CD28 stimulation as we expected, some that had both IL-2 and IL-15, perhaps at different concentrations, one that only had IL-15 and one that only had IL-2. It's important for me to point out here, we weren't looking for the ideal or identify an ideal reagent here. We were actually looking to validate our platform that could be used to identify reagents that were ideal for a specific phenotype in manufacturing. So our first order of business was actually using these MCBs to manufacture CAR T cells from diffuse large B-cell lymphoma patient T cells. And so these are patients that would be eligible for CAR T cell manufacturing and treatment. And so we were able to obtain these samples and manufacture CAR T cells using either Dynabeads plus IL-2, which is always denoted as DB, or our 4 different MCB formulations. And so we can see that they have similar levels of CAR T cell transduction across these different conditions, but we have superior CD8 to CD4 ratios with some of our MCBs, and significantly more CD62L expression using our MCBs compared to Dynabeads. And CD62 is a marker of memory and naive T cells, which is associated with superior clinical responses. Next, if we look at expression of triple exhaustive markers, we can see that compared to the Dynabeads, we have less expression of triple exhausted cells or triple expression of exhausted markers, indicating that these cells have superior baseline characteristics. We then did multi-dimensional mass cytometry on these cells to look for markers of self-renewal, and we can see if we look across the different clusters in the maps, generated by our CyTOF analysis, we can see that there are certain clusters that are being enriched for within our MCB products. And we want to highlight actually cluster 10, which is here in red. And you can see these are with markers associated with memory and self-renewer. And in Dynabead, this is this cluster here. But you can see with our MCB formulations, this cluster is increased. So, and then we look at TCF1 expression, which is again associated with self-renewing CAR T cells. We can see our MCB manufactured CAR T cells have significantly more expression of TCF1 compared to Dynabead plus IL-2 manufactured CAR T cells. We next wanted to look at how our MCB manufactured CAR T cells looked metabolically, because we know that as T cells differentiate and they lose stemness, they actually move from oxphos metabolism to glycolysis. And so we wanted to see how our T cell products were faring. In order to do this, we used a seahorse assay, which looks at basal respiration and spare respiratory capacity. And so this graph here, or this diagram here, shows the different things that are measured by a seahorse. So we have basal respiration, and then an inhibitor added, which decreases ATP production, and then you have another inhibitor added, which causes the maximal respiration. And then once you add the last inhibitor, it gives you the spare respiratory capacity. And so we could see using this assay, our MCB manufactured CAR T cells has had improved basal respiration and improved spare respiratory capacity. So finally, we wanted to test these cells in a mouse model of lymphoma. And so using NSG mice, we injected, IV injected Raji cells and treated them 4 days later with CD19 targeted CAR T cells. And we imaged the mice and collected blood. And as you can see, at 31 days post CAR T cell treatment, our Dynabead treated mice actually are showing signals of disease, where our MCB treated mice don't have any signals of disease. If we look at the survival curve, we can see that our MCB manufactured CAR T cells, which are in here, have improved long-term survival compared to untreated or Dynabead treated mice. We had a few mice that were long-term survivors in the MCB treated conditions. And so if we look at day 188 post first tumor inoculation, we can see that these two mice still had circulating CAR T cells in their periphery. And so we decided to re-challenge them to see if these T cells were still functional. In order to do this, we, again, at day 188, we saw that they had T cells. We re-challenged them with half the dose of Raji tumor cells, and then we IVIS them. And as you can see, compared to a naive new control of mice, the MCB treated mice still were able to control the tumor almost 200 days past or after their first tumor challenge. And so this is indicated here, where they still have very little tumor burden and then had improved overall survival. So finally, we had done all of these studies in lymphoma and lymphoma models. We wanted to see if our multi-cytokine backpacks could also produce CAR T cells with superior phenotypes against solid tumors. And so for these studies, we inoculated SCID beige mice with SKOV3, which is an ovarian cancer cell line, and we treated 7 days later with our either Dynabeads or multi-cytokine backpacks. And as you can see from these imaging images, the untreated mice died by day 36, but the tumor burden of the Dynabead treated mice is far greater than the ones of some of our MCB treated mice. And if we look at the survival curve, you can see there's significant differences between how our long-term surviving MCB treated mice and the Dynabead treated mice. And so in summary, we think that this platform can be used for a variety of different methods. And so as I showed you, our library was able to create particles of diverse formulations. And then we were able to test this in a 96-well format in which we identify statistically superior multi-cytokine backpacks that were functionally superior, and then they had memory phenotypes, evidence of self-renewal, metabolic fitness, and enhanced in vivo efficacy. We think that this platform can be used for future applications to identify ideal manufacturing reagents and can be modified to address different questions. And so we can identify ideal signals for manufacturing, such as we did here, where we looked at cytokines and antibodies for producing less differentiated CAR T cells, but we could also optimize CAR T cell phenotypes based on the tumor type, such as optimize them for certain expressions of chemokines or chemokine receptors for solid tumors, and also apply this to improve other types of cellular therapies or improve the ease at which you can manufacture CAR NK cells or CAR gamma delta T cells. We also think this could decrease the cost of manufacturing, and as I mentioned already, we can apply this process for solid tumors and other types of cell therapies. We also had some very interesting data that came out of our spent media analysis, and we think that there's a lot of work still to be done in this arena, and so we could definitely optimize cell culture medias of our cell therapies for cell manufacturing, and we need to delve more into understanding the metabolism and nutrient needs of CAR T cells during manufacturing so that we can produce superior CAR T cell products that perform better in patients. With that, I'd like to thank my lab and especially my brilliant graduate student, Heather Lynn, who led most of this work in collaboration with Biagio Uricoli from Eric Dreeden's lab, who we collaborated with heavily on this project. I'd also like to thank 908 Devices, especially G. Anderson and Mila Neffling, who helped us with the Rebel analysis and technical help with that. I also would like to thank our collaborators at Emory, our funding, and the course that we use, and I'd be happy to take any questions. Thank you.
So indeed, now it is time to go to our questions, and it is time for you to take part in our session. So thank you so much, Dr. Rafiq. This was really excellent. And for our audience, please type in your questions in the questions box in your GoToWebinar control and dialog window, and we will start our questions session here. Our first question is around the beads used in the cytokine delivery. Thanks for the fascinating talk. I am not sure I understand the logic behind why the bead-bound cytokine delivery is qualitatively different from soluble cytokines. Why would this result in less differentiated phenotypes?
So one of the advantages, there's a number of advantages for this platform, especially bead-based cytokine delivery. One is we can actually control the amount of cytokines to a very low level the cells are receiving. Secondly, it might be during manufacturing, save costs on the amount of cytokines that are being used. And then lastly, certain cytokines such as IL-15 actually are ideally used by T cells when they're presented through the IL-15 receptor. And so we think we also might be getting superior delivery of cytokines using the speed-based approach.
That's excellent, and it's all very condensed, right? So it's interactions are guaranteed. So there's another question here on the multi-cytokine backpacks. Are they available for research or purchase?
So right now we're doing preclinical development of them in my group, and so we're happy to collaborate with anyone who would be interested in helping us expand this platform. But currently they're not for commercial use.
So from there, these are not yet available for everyone. But which applications do you see next for this platform? What are your brilliant graduate students busy with or planning to be busy with?
Yeah, so there's a number of ways we can expand upon this. Obviously, we'd be interested in looking at this for translation to patient applications. So that would be the first thing on our agenda. But in terms of the platform, I think this is could be a really powerful platform for manufacturing difficult to produce immune cells, such as CAR NK cells or CAR gamma delta cells. And so because we can add or optimize even the cytokines that are being provided to the cells during manufacturing, we think we can maybe shorten or improve the manufacturing process for these types of cells. And so that would sort of be the next approach that we'd like to take with our platform.
Now, we do have a bit longer question, which is a follow-up question for the beads versus soluble protein. the question reads, have you or do you plan to measure the actual amount of cytokine seen by the cell, whether on your beads or in soluble forms, for example, by measuring signaling or phosphorylation? So how deeply have you characterized?
The beads, right. So we did characterize how much cytokines are on all the beads. If you remember the three-dimensional plot where we showed the different amounts of IL-2 and IL-15 antibodies that were on the beads by fluorescence. And so we have quantified, we do have an idea of how much cytokine that we're providing. I think there's some interesting work that could be done around whether the particles get reloaded with cytokines that the T cells are maybe producing and what the on-off rate could be. But in terms of quantifying it, we've done it in that way. We could also look at the signaling within the cells. That could be an interesting approach too, where we can look at the amount of signaling that's actually going in inside the CAR T cell.
Excellent. So there's still quite a bit to do with understanding all of this and with improving the manufacturing process overall, perhaps with some cell culture media improvements along with all the biology. It's so fascinating. I do believe it is time for us to close our session. Please do not hesitate to contact us for further questions.